open. Okay, I'll start sharing my screen and we can start with our session now. Are my slides visible? Just can anyone quickly confirm whether my slides are visible or not? Okay, so good evening all. So we'll start with our problem solving session for week 9. So the topics that we will be covering today are multiple hypothesis testing correction, FDR correction and interpretation of DGE analysis result, functional enrichment analysis, RNA-seq data analysis hands-on 2 and hands-on 2 setting up the system. So the course Next Generation Sequencing Technologies Data Analysis and Application is being taken by Professor Riddhiman Dhar from IIT Kharagpur. So I'll quickly introduce myself as well. I am Pooja Yadav and I am a PhD scholar as well as a PMRF fellow in Indian Institute of Technology, Bombay in Department of Biosciences and Bioengineering. I am a PhD student in molecular virology lab. So we'll start with our session now. So again, like previous sessions, we'll have questionnaires. So first, you please try to answer the questions, and then we will discuss what are the concepts for those questions. So, but please try to answer the question first so that session would be more interactive. So we'll start with our question number 1. Uh, The question number 1, it states that let's say in a multiple hypothesis testing, the number of statistical tests are 50. And the probability of making one type 1 error is in this multiple hypothesis testing is 0.395. What is the probability of type 1 error for each statistical test? So please remember type 1 error is false positive, and if we are doing 50 statistical tests, the probability of false positive among all the tests is 0.395. We need to calculate the probability of false positive for each test. So please try to solve this question and try to answer this question as well. We have one answer B. Is there any other answer that might be correct apart from B? Okay, let's look into the concept. So the correct answer for this question is B only. So we will look into the concept. So in the question, so the probability of not making one type 1 error is 1 minus the probability of type 1 error for each statistical test. So for not making type 1 error is 1 minus probability of type 1 error for each statistical test. So what would be the probability of making one type 1 error? It would be 1 minus the probability of type 1 error and to the power the total number of statistical tests. So in this question, what is given? We are given with the total number of probability. The probability of making one type 1 error when we are doing 50 statistical test, which is 0.395. And if we compute this value with the formula and then we solve this equation, then we would get the probability of making one type 1 error for each statistical test. It would be 0.011. So the correct answer for this question would be B, 0.011. So basically what is given, we are given with total number of statistical tests and the probability of making one type 1 error. So we need to in total of 50 statistical tests, and we need to compute the probability of type 1 error in each of the statistical tests. So the probability of type 1 error when we are doing 50 statistical tests, it would be 0.395 equals to minus 1 minus probability of type 1 error to the power the total number of statistical tests. And when we solve this equation, then we will get the probability of making one type error for each of the statistical tests. So I hope this question is clear. So we will move to our next question, which is question number 2. The question number 2, which states that which of the below options are fewer FWER correction method? A is Benjamini-Hochberg correction, B is Bonferroni correction, C is Story-Tibzerni correction, and D is Home-Bonferroni correction. Please try to answer this question. Okay, we have one option B and D. the correct option for this question is B and D, which is Bonferroni correction and home Bonferroni correction. Okay, so okay, just let's look into the concept for this question. So the family wise error rate, it refers to the probability of making at least one type 1 error which is false positive when performing multiple comparison, or when we are doing multiple statistical tests to reject our null hypothesis. So this correction method aim to control this overall error rate to ensure that likelihood of making any false discovery is minimized. So why we do this? Uh, correction method? So just in case we need to be sure that the positive we are getting are true positive and not the false positive. So here we are minimizing the type 1 error, which is we are minimizing the outcome of false positive results when we are doing the statistical test. So in the first option, which is given Benjamini-Hochberg correction, so this method, it is designed to control the false discovery rate. It is not designed to control the false positive rate. So this is not the correct option. Whereas with the B option is Bonferroni correction. So this is basically the classical method used to control the family-wise error rate. So how it is being done? So it is being done by dividing the significance level, which is alpha here, by the number of tests that are being performed. Let's suppose we have alpha, our significance level, which is alpha, and we have n number of statistical tests. So we divide alpha by n. So what it does, it it is a very stringent way to minimize the false positive outcomes in this case. So this option is correct. The third option is, again, it is FDR, false discovery rate method, but not the not nullifying the false positive. The D option is home Bonferroni correction. Home Bonferroni correction is again the modification of Bonferroni correction. So in Bonferroni correction, what happens is there is increased chance of false negative as well, because it is a very stringent method. But in the modified version of Bonferroni's home Bonferroni correction, where a step uh, it is improved upon the Bonferroni correction by applying adjustment in a less conservative manner. It means it is a comparatively less stringent than Bonferroni. They give order to the p values that they are computing, and then they come to an output whether it is a false positive or a false or it is not a false positive. So it is a slightly flexible than the Bonferroni, but uh, they both are these both methods are used to control the family wise error rate. So the correct option would be B and D, because both of the methods are for family wise error rate correction method. So you guys can look into each of these methods, so how the probabilities are being calculated for each of the methods, and how where the false positive rates are being computed, where the false discovery rates are computed, and how they are impacting our analysis, how they are and based on these methods, how we are nullifying our null hypothesis, or and how we are saying that the result we are getting are the true positive and not the false positive. So each method has their pros and cons, so you can go into each of the methods and try to know about them more. So I hope this question is clear. So we'll move to our next question. Which of the below options are true? Please go through this question and try to answer this question. So we have one option which is B and C. We have another option, just B. Is there any other option that might be correct apart from B and C, or including B and C? Okay, let's look into the option. The correct answer for this question is B and C, uh, which is FDR allows a certain number of false positive. C is Bonferroni correction, so shows more type 2 error than home Bonferroni correction. So let's look into the concept. Okay, we'll just uh, look into how these are related to type like type 2 or type 1 error. We won't go into all the formulas and all, we'll just go into the concept of each of these methods. um, the first option was fewer allows false positive. As we have studied in the previous question as well, they have a very stringent method for neglecting or for removing the false positive. So it is designed in such a way that it is minimizing the possibility of one false positive. So the first option is false, but in this case what happens is the type 2 error increases, the probability of type 2 error, which is false negative, also increases. So it is basically a trade-off for avoiding the false positive. So the B option was FDR allows a certain number of false positive. This statement is true because the false discovery rate methods allow for some false positive, but it controls the proportion of false discoveries among all the rejected hypotheses. Basically, this is a more lenient approach than that of a family-wise error rate, where it allows few of the false positive, uh, but it also is giving the false discovery rate, because it says that the rate at which these false positive are coming. So this again gives a parameter, extra parameter, which is false discovery rate. So we can compute that whether based on the total positive, we can say that this proportion is false positive among all the positive that we are getting. So this approach is more lenient than family-wise error rate and is useful in cases where it's acceptable to have a control number of false positive, especially when dealing with large number of comparison. So this option is true. The C option is Bonferroni correction shows more type 2 error than home Bonferroni correction. So it is also true that we, in the last question as well, we saw that home Bonferroni was a modified version of Bonferroni to decrease the type 2 error, which is a false negative. So Bonferroni was more stringent than the home Bonferroni correction, but the home Bonferroni, it gives a lesser number of a type 2 error in comparison to the Bonferroni. So which is also, the C statement is true. The D statement was FDR can control type 2 error, which is false. Because the false discovery rate is primarily concerned with the type 1 error, which is false positive. It does not tell about the anything about false negative. It tells you about the false discovery rate, which is basically it is directly or indirectly telling about the false positive only. So the fourth option is also false. So the correct option for this question would be B and C. FDR allows a certain number of false positive, and C is Bonferroni correction shows more type 2 errors than home Bonferroni correction. So I hope this question is clear. So we'll move to our next question. Question number 4. Let's say a bin contains 20 balls within which 12 balls are red, representing the pathway of apoptosis, and 8 balls are green, representing oxidative phosphorylation. Now if you draw 3 balls randomly from bin, what will be the probability of being 2 balls from apoptosis and 1 ball from oxidative phosphorylation pathway? So basically 2 red ball and 1 green ball. So we have a 20 balls which is divided into 2 parts, 12 and 8. 12 are red ball, 8 are green ball. So now we are randomly drawing balls from the 20 balls and we got the 3 balls. And in this question what is asked, what is the probability of 2 balls being the red balls and 1 ball being the green ball? Please try to answer this question. Please take a few minutes and try to solve this question. has anyone solved this question? Okay, let's look into the concept for this question. So, in the information, we are taking the balls without putting it back. So, it is basically follow the hypergeometric rule, which is when we are consistently taking some samples from a particular, from a consisting sample size. When we are taking out a random sample from a consisting sample size, so it is follow a hypergeometric rule. So among 20 balls in the question, 12 balls that were red are from apoptosis pathway, and the 8 balls which are green are from oxidative phosphorylation. So in the question what is asked, so if we are drawing the 3 balls randomly, what is the probability of 2 ball being red and 1 ball being green? So hence the probability of 2 balls from the apoptosis pathway which are red and 1 ball from the oxidative phosphorylation which is green. If we draw 3 balls randomly from the bin, consist of 20 ball, the probability would be the which is given. This question, what is missing? Something is missing. Okay, right. The probability of 2, the red from 12 balls, which is, and the probability of 1 from 8 divided by the probability of 2 among 20. So if you divide 2 by 12, 1 by 8 and 3 by 20 and we compute this formula, then we get the probability equals to 0.0586. So the correct option for this question would be I'll just 0.058. So this is not the correct option. The correct option is 0.586.
Ma'am, please can you explain it again?
Yeah. So here, if you compute, so we have two, we are taking 3 balls, right? So, and what is asked in the question, 2 balls we are taking red and 1 balls we are taking green, and out of uh, 20 we have 12 red ball. So when you divide 12 by 2 by 12 into 1 by 8, so because we have one green ball out of 8, and then we divide it by, we have a total of a 3 ball divided by total number of balls. So let's calculate it here only. There is some misunderstanding in the question. I'll just calculate quickly and tell. Okay, this question has some issue. So the calculation here is wrong. So we'll get back to this question. If not in this class, we'll definitely get back to this question in the next class. There is something wrong in this question, so we'll definitely get back to this question. Okay, so let's move to our next question. Multiple hypothesis testing increases. So please go through this question and try to answer this question. We have one answer B. We have another answer B. The correct answer is B only, true positive. Um, Sorry, the correct answer is uh, not B, it's D. So it false positive. When we have a multiple hypothesis testing, so the p value also increases. So if the p value also increases, it gives the more number of false positive. So that is why we had those stringent test methods like Bonferroni test, were to compute, which were to nullify these discovery of false positive. So, but when we do this basic multiple hypothesis test and we go for the p value, then we have the increased number of false positive. So, the correct option would be D. So, in the previous session, we have seen what are the type 1 error and what are the type 2 error. So, we won't again go to this.
Ma'am, the correct option is the false positives, right?
The correct option is the false positive, D.
Yes, ma'am.
Yeah. So, let's move to our next question. Uh, the question number 6, it states that in a differential gene expression analysis of 10 genes which are given here, one got 10 different p values, which are these. So these are the p value for each gene. What will be the q value of gene 7 according to Benjamin-Hochberg correction? So Q value is the basically the false discovery rate only. The FDR cutoff is 0.1. So please try to solve this question. have one option which is 0.0585. Is there any other answer that might be correct? Okay, let's try to solve this question. Um, yes, both the answer are correct. 0.059, which is, so what are we are given with? We are given with differential expression of 10 genes, and we are given with the p-value for each of the genes. So for again Benjamin Hochberg, we need to place these genes uh, based on the p-value. So the most differentially expressed gene, from least differentially expressed gene to most differentially expressed gene. So when we place all the genes based on the p-value, then we get uh, gene 1, 2, 9, 5, 4, 8, 7, 3, 6, 10. So these order are based on the p-values, the ascending p-values, based on their differential gene expression. So in this question, what is asked? What will be the Q value for gene 7 according to Benjamin Hochberg? So here at the seventh rank we have gene 7 only. So for to computing the Q value for gene 7, we need to we have the gene expression, which is the p value for our gene expression, which is 0.014 into number of genes divided by rank of genes. So total number of genes we have 10, when we divide it by the rank of gene, which is 7. So we get the value 0.058 for this question. So the correct
Ma'am, I have one doubt.
yes.
So, ma'am, in our question, it is written to compute the Q value of gene 4. So, if we are taking gene 4 in consideration, then the rank will be 5, right?
The rank will be 5. Yes.
And the value will be 0.0158 according to the question given.
Yeah, the value here, the value would be changed. These two values would be changed. So based on that, the answer would be according to that.
Ma'am, what will be the value then for the Q value to compute? Like in this case, we for gene 7 we took 0.041.
Yes,
So the value for G4 will be 0.0158.
it will be this 0.4 will be 0.0249.
Oh, thank you, ma'am.
Yes. So let's move to our next question.
So, ma'am, another question, ma'am. Sorry
Yes.
to interrupt. Uh, Means how do we know the these values? I can't understand that.
Like which values?
Um,
So
that
according to
0.04.
okay, so according to Benjamin Hochberg correction, so you have, like, there are ways to compute the p values. They have some like, I mean, uh, so in Bonferroni, we have seen that they divided by alpha upon n to calculate the p-value, and based on that, they are given with certain parameters. Based on that, how many statistical tests they have done, what is their error rate, or what is the alpha. So based on that we calculate these p values.
Yes, ma'am. Another question is given, that question number 7. The question is, how many non-significant genes are there? Then how to know how many significant and non-significant genes are present in such type of questions it comes?
So that is why we are calculating the p values, and we are giving the how many are the false positive. So we are nullifying our null hypothesis. So null hypothesis is what it states, that these genes are not differentially expressed. So how we are saying that, based on our p values we are saying that. So for those we are nullifying our null hypothesis, these genes are not significant.
Yes, ma'am. So,
Yes.
ma'am, in this case, as I talked, like in if we want to compute Q value for gene 4, the value will be 0.0249.
The value would be yes.
Thank you, ma'am.
So as the p value increases, so the so here these for these genes the p value is comparatively higher. So like this they are not significant in our when we do this differential gene expression, these genes are not significant for our analysis. So based on the p values we compute that which genes are significant and which genes are not significant. So let's move to our next question. So, in which of the below mentioned analysis a rank list of gene is given as input? A, gene over representation analysis. B is gene set enrichment analysis. C, both A and B. D, differential gene expression analysis. Please try to answer this question. So in this question we are giving a rank like we have seen. So over here it is again asked, in which below mentioned analysis a rank list of gene is given as input. So what it why do we give the rank for genes? What based on what we are giving the rank of genes? So based on this, please try to answer this question. We have one option D, which is differential gene expression. Is there any other option that is correct for this question? Okay, let's look into the concept for this question. The correct option for this question is B, gene set enrichment analysis. Let's look into why. So gene over representation analysis. So in this type of analysis, we typically provide a list of genes that are either upregulated or downregulated, but they are not ranked. So the analysis check whether a certain biological pathway or a function are overrepresented in a gene list compared to what we expect by chance. So over here, so we are the um, giving the information about the upregulation or downregulation of the gene based on which a pathway is, whether a pathway is overrepresented or not. We are here not giving a rank to the genes based on anything. So while the option B is gene set enrichment analysis, GSEA, so it is designed to take a ranked list of genes based on some measures which are expression level or differential expression p value. So in then assess whether predefined gene set like pathways are they enriched at the top or a bottom of this ranked list. So for this, for gene set enrichment analysis, so we are giving a rank to the gene either based on their expression level or the differential expression, or basically for their p values. So then we say that whether the predefined gene set they are enriched either at the top or the bottom of this rank list. So top or a bottom is whether they are differentially expressed or not differentially expressed. So the fourth option is differential gene expression analysis. So this analysis it identifies genes that show statistical significant difference in expression level between different conditions. So here for this we already have come to this step that here we know that these genes are differentially expressed. So based on these enrichment analysis and other statistical tests, we here now know that these genes are differentially expressed. So again here we are not giving any rank to the genes, but only here gene set enrichment analysis we are giving the rank of the genes based on the p values, and these p values would further be useful for computing the differential gene expression. So the correct option would be gene set enrichment analysis, which is B. So I hope this question is clear. So let's move to our next question, which is question number 8. Gene A shows a difference in expression between disease and healthy sample. The threshold for significance is set to 0.05 and the number of genes that have been compared between two samples were 5000. So according to Bonferroni method, what should be the p-value of gene A so that the null hypothesis can be rejected? So the null hypothesis, gene A doesn't have a difference in expression between disease and healthy sample. Please try to solve this question. Please take few minutes and try to solve this question. Has anyone got the answer for this question? Please take few minutes and try to solve this question. Okay, let's look into the answer for this question is. So in this question, what is given, we are given with the p values that we have done uh, some statistical test for 5,000 genes, and we were found with that gene A was differentially expressed, or it had a difference in expression. And for this p value, so the criteria, so based on the p value which is 0.05, so normally we take 0.05 as our p value for computing whether we can reject our null hypothesis or we cannot reject our null hypothesis. But in this question, so we need to now calculate the new p value according to Bonferroni method, so whether we can reject our null hypothesis or not, whether, or we can say that gene A is differentially expressed or gene A is not differentially expressed. So for the p value which is given is 0.05 and the total number of genes are 5,000. So according to Bonferroni method, we have new p value. So in the first question we have seen that the new p value is alpha by n. So here we are given with the alpha which is 0.05 and divided by n which is our total number of genes. Here in this case which is 5,000. So the null hypothesis will be rejected. So this is our the new p value. So when we compute this, we will be getting 1 into 10 raised to power minus 5, which is our new p value according to Bonferroni method. So here you are seeing that this p value is very less. So again, according to Bonferroni method, it is very stringent method, so it does not compute any false positive, because here you can see the p value is very less. When you compare the p value to this value, it is very less, but when we have such stringent method, we tend to get more number of false positive. More number of false negative. So there might be some genes that are actually differentially expressed, but when we go with the Bonferroni method, we tend to lose those genes, and the number of false negative also increases. So anyways, the correct answer for this question is A, which is p value less than 1 into 10 raised to power minus 5. question is clear. So let's move to our next question. Which of the following options are true regarding gene ontology? A is it's a database of functional annotation of genes. B is biological process is one of the sub-ontologies. C is partially hierarchical. D, parent nodes can describe more specialized function than daughter nodes. Please try to answer for this question. We have one option A, B and C. The correct option for this question is A, B and C. So the gene ontology, it is a database for functional annotation of genes. So biological process, these genes are involved in such many biological process. So biological process is also one of the sub-ontologies. So it is partially hierarchical, which is also true, and parent node can describe more specialized function than daughter nodes, which is not true. So the correct option for this question would be A, B and C. Let's move to our next question. Which of the following databases are used for functional enrichment analysis? A, the Cancer Genome Atlas, the Molecular Signature Database. C is Kyoto Encyclopedia of Genes and Genome, KEG. D is Ensemble Database. Please try to answer this question. So there are few databases where you can find these either the RNA-seq data or mostly the enrichment analysis data. So which of the database where you can find such data for functional enrichment analysis? These database has already a certain information about where these genes are differentially expressed or not. So we compare our data with their data as well to make sure whether it is correct or not. So uh, so one is molecular signature database and the other is Kyoto Encyclopedia of Gene and Genome, which is KEG. So I'll share these two links in the chat box. Please have a look for these database. Just take a few minutes and have a look for these database, or you can go to these database later on as well. the molecular signature database is a resource of tens of thousands of annotated gene sets for use for uh, GSEA software. The other one is KEG. Again, KEG is a database resource for understanding high level functional utilities of the biological system. So, if you go, so you can go through these databases and try to navigate how these databases stores the data, and you can go after the class as well. So let's move to our next question. So the correct option for this question would be B and C. So let's go to our next question. Question number 11. Which of the below options are true about the calculation of enrichment score? Uh, BA is test whether the member of a gene set for a particular pathway is present at the top or the bottom of the ranked gene list. B, the score decreases if the member of a gene set for a pathway is found while traversing through the ranked gene list. C is the final ES score is the maximum deviation from 0 while traversing through the ranked gene list. D is rank gene list is not needed for calculation of ES score. Please try to answer this question. We have one option A and C. A and C are the correct options. So the we give the enrichment score based on different values. One is the difference in expression of difference based on the p values as well. So what is tell that we have a specific gene, and we from these ES score we can say that whether this gene set for a particular pathway is present at the top or the bottom of the rank list. So for the top we have the highest enrichment score, for the bottom we has the least enrichment score. So we can navigate based on these scores, we can navigate whether the enrichment of our gene of interest is at the highest or where it lies among the highest and the lowest. So the score basically is the maximum deviation from the zero. It does not decreases if a member of a gene set for a pathway is found while traversing through the ranked gene list. It actually sort of increases, not decreases. C is the final yes score is the maximum deviation from a zero while traversing through the ranked gene list, which is true. The option is ranked gene list is not needed for calculation of ES score, which is false. We need the rank gene list when we calculate the ES score. So, okay, I'll share a very good paper. So, how you can calculate these ES score? There are different ways. So, you can go through this paper after the class. So, in this paper, the gene set enrichment analysis a knowledge-based approach for interpreting genome-wide expression profile. So here it's a very good paper. So they have given with different ways how you can do the statistical analysis and how you calculate these enrichment scores, and you can, based on this, you can do the multiple hypothesis testing. So it's a short paper only, you can go through this paper after the class, so you can have a better idea about this. So how we do this gene set enrichment analysis, what are the steps? So we basically rank all the genes based on the p-values. Then we calculate the enrichment score for each predefined gene set by determining if gene from the set appear disproportionately at the top or the bottom of the ranked gene list. Then we again normalizing the enrichment score to make gene sets comparable by adjusting the size of the gene set. So we need to do the normalization before any analysis, so then we can compare, we are at the same level of comparing for the different sets of samples, or a different, if these samples were from the different time points. So we need to normalize our data before doing any analysis. So the correct option for this question would be A and C. Test whether the member of a gene set for a particular path is present at the top or the bottom of the rank list. And the final ES score is the maximum deviation from 0 while traversing through the ranked gene list. So we'll move to our next question, question number 11. In gene set enrichment analysis, calculating NES normalized enrichment score help to compare the result across gene set. By normalizing the enrichment score, GSEA remove the account for biases in gene set size. A option is assertion is false but the reason is true. B is assertion is true but the reason is false. C, both are true. D, both are false. Please try to answer for this question. We have one option which is C, both are true. Again, we have C option. So is there any other option that might be correct apart from C? Basically, the C option is correct only, as in the last question we have seen that we need to do the normalized enrichment score before we do any analysis. So basically it removes all the biases, either it is coming from sequencing or coming from time point or gene set size, etc. So both are true. The C option is true. So GSEA is a method used to determine whether a predefined set of genes shows a significant difference between the condition, disease versus healthy. So NES is calculated by adjusting the raw enrichment score to account for gene set size, ensuring that results are comparable across gene set of different size. So we do this normalization in order to account any biases coming from the gene set size. We need to be ensured that these gene set size are not like is not responsible for any false positive. So we need to normalize our data first. This normalization, it removes biases, allowing for fair comparison. So the correct option is C, both are true. So I hope that is clear. That was our last question. So there was a confusion in one of the question, which is question number, I think this question number this question number 4th. So we'll come to this question in the next class. We'll start our next class with this question. So apart from that question, is there any doubt regarding this lecture or any other doubt?
Hello, ma'am.
Yes.
Ma'am, I have one question, ma'am. In that, we are calculating Q value, no, ma'am, based on.
You are not audible clearly. Can you speak a little bit louder?
Ma'am, we are calculating Q values, no, ma'am, based on
Yes.
Benjamin Hodge.
Yes.
Ma'am, after that, how to count the number of non-significant genes, ma'am, based on FDR?
So we are getting this FDR. We are basically calculating the false discovery rate. Right. When we know the false discovery rate, we can, from among all the positive that we are computing the differential gene expression, now we have a false discovery rate. Then we can compute that these number of genes, they might be false positive. They might be like false positive for our data.
Ma'am, in our question, they are asking total how many non-significant genes are present, ma'am?
Total how many non-significant genes are present? Okay.
Yes, ma'am.
So where was
How
this
to count,
question?
ma'am, based on Q value or based on FDR cutoff?
Okay. What will be the Q value for a gene according to if their cutoff rate is? Okay. So in this, what was the question asked for you?
There are two questions, ma'am. First question was what will be the Q value of gene 4 according to the Benjamin Hodge correction?
Right.
And second question is how many non-significant genes are there?
How many non-significant genes are there? Okay.
Yes, ma'am.
So we have the Q value as well as FDR. Okay. So based on, okay, I'll have to check for this question as well because I did not look into it. Can I come with this question like in the next class if possible?
Okay, ma'am.
Yeah, so we have now two questions that we can start discussing these two questions in our next class. Then we will move to our next session. So we'll start our session with these two questions and then we will move to our next week problem solving session. So I hope so. I know answer, but I don't want to be like, I want to be sure that I am like giving the correct answer, so there might not be any confusion. Okay, so if there are no any other doubts, we can wrap up the session and we'll meet for our next session the next week at the same time. So you guys can leave the meeting. Thank you all for joining in.